been interested in plants since an early age, in particular how they can grow and develop in such diverse conditions. Grafting is an incredibly clever process, and I love that grafting has been done for thousands of years, and now we can take modern tools and techniques to better understand how plants do this incredible process. My name is Charles Melnyk, and I'm a professor of plant physiology. My research focuses on the process of plant grafting. Grafting involves the cutting and joining two plants together to make a superior plant, to take the best properties of each plant. It's been practiced for thousands of years, yet we have little idea of how plants actually graft. My research focuses on trying to understand the process of plant grafting, how plants heal wounds, how they regenerate tissues, and how they can actually connect. It also focuses on the darker side of plant grafting, the process of parasitic plants, whereby they can grow and connect to another individual to withdraw nutrients. The goal of my research is twofold. I want to understand how plants graft from a fundamental point of view. I want to understand the genes, the pathways, and the proteins that are involved in successful grafting. Secondly, I want to take this information and use it to improve Swedish forestry and Swedish agriculture. I want to graft new combinations of plants that improve stress tolerance and disease resistance and allow plants to grow better and stronger. What motivates me is working as a team and making new discoveries. It's incredibly exciting and rewarding to discover things that have never been found before, while at the same time mentoring a next generation of scientists, including researchers, postdocs, and PhD students.
Welcome, Charles.
Thank you. Nice to be here.
Nice to have you here. Well, as you said in the video, your research is all about grafting, which is a clever gardening technique where parts from different plants are joined together to grow as one. And this is called ympning in Swedish. In nature, this sometimes happens on its own, for example, when roots or branches meet under the bark and naturally fuse. Why do you think some plants have evolved the ability to graft like this?
Yeah, so I mean, we don't know why plants can graft, but of course it's an incredibly important and interesting ability, and it probably has to do a lot with their ability to heal wounds so efficiently. So plants are really good at regenerating. So some plants, when you cut them or chop them into pieces, they can grow completely new pieces. And we think that the grafting ability probably relates to this, so that when you wound them or you cut them, they can actually reconnect and regrow and regenerate really, really well.
And why can't animals graft?
Yeah, wouldn't it be cool if we could graft animals? And, you know, I think there's a couple things going on. Of course, animals are also good at healing wounds, but it seems that many plants are just much better than this. When they get a very, very deep wound, humans need help. They need microsurgeons to reconnect their pieces, but plants don't need this. There's also something different about the immune systems. So even when you do a blood transfusion, for example, there's issues with making sure the blood is compatible and everything is okay. But plants don't seem to have this. As long as the plants are closely related, they can reconnect. They don't have issues with immunity or their immune systems, and they're happy to grow together as one.
So exciting. Well, a lot of the vegetables that we eat have actually been grafted, which I didn't know before you told me. How common is grafting in growing the vegetables that we find in the store today?
Yeah, so I think grafting is, of course, maybe it's best known for the fruits that we actually eat. So when you think of like apples or pears, cherries, peaches, oranges, they're mostly completely all grafted. The wine that we drink is all from grafted plants. But vegetables, of course, is getting very, very popular now and common. So when I think of grafted vegetables, tomatoes, cucumbers, melons, watermelons are, for the large part, being grafted, especially when they're grown in greenhouses, and it's getting more and more common.
And then what is a goal with this grafting?
So the goal is to make plants more disease resistant, more climate tolerant, and to improve their yields. And I think they do this by combining the best of two plants.
Well, why don't you just get prepared for your lecture and tell us more about grafting?
Okay, thank you.
And I would like to, please. I would like to introduce Charles Melnyk, professor of plant physiology, and he's going to talk about connecting plants to friend or foe.
Thank you. Okay, great. So for the next 15 minutes or so, I'd like to present a little bit about the research that I've been doing. And of course, I think the video is also given a nice introduction. But my research, for a large part, has focused on the process of plant grafting and parasitic plant infections. Now, one thing that's very interesting about the process of grafting is that it's been practiced for thousands of years. And how do we know this? We can actually go back to antiquity. We can look at the Bible, we can look at other sources, we can look at Roman mosaics from 2,000 years ago, and we actually see people talking about grafting and performing grafting. More recently, maybe 500 years ago, we also have texts that describe different types of grafting and how plants can be grafted. And we can also see this today with modern grafting. So the same techniques that were practiced 500 years ago, we still see evidence of this. So cutting plants, joining them together to form new combinations of plants. So why are we actually grafting plants? And I alluded to this a little bit earlier, but one of the major reasons that we actually graft plants, and this was done probably thousands of years ago, this was the reason, is that we wanted to clone plants. And what does it mean to clone? It means to make genetically identical copies of the same plant. You discover a delicious apple tree, you want to make more of it, so you actually use grafting to do this. We also use grafting to increase yields, increase stress tolerance, and also modify the size of plants. And this goes back to the idea that if we take two different plants, one has a great root, the other one has a really great tasty fruit, we can graft them together to get a superior plant. So nowadays, our use of grafting is growing. This is becoming more and more common. About a third of our top agricultural plants are being grafted, contributing to markets worth hundreds of billions of euros each year. And there's an estimate of over 1 billion plants are being grafted each year, which is a huge, huge number. So I'd just like to very briefly present two examples to you about how grafting has changed horticulture and agriculture. One is the example of apple trees. And I'm showing you three different pictures here, taken different times. And in theory, they could be the exact same apple variety. This could be like your Granny Smith apple. And when we look back a couple hundred years ago, before they used grafting, the apple trees were quite large. They could grow 5, 6, 7 meters tall. And people realized that we could start grafting on a root of a different type of apple that would cause the apple trees to become much smaller. And this was known as dwarfing. So you can see here, when you look at your garden in Sweden or in Uppsala, you typically have trees like this. And this is an intermediate dwarfing. So you get a different root, you make the tree a little bit smaller. But in modern apple orchards, they actually don't look like this anymore. They look like the image on the right. So here the trees have extremely dwarfing rootstocks and they look more like a hedge. So an individual tree produces much fewer apples, but the productivity overall goes way up 2 or 3-fold higher than not having these hedges, and it also makes it much easier to harvest. Another example is with our grapevines. So this is again for the wine industry. And about 150 years ago, an insect arrived into Europe, which was called phylloxera, and it started to attack the European grapes. And they had no resistance to this insect, so it destroyed grapevines in France, in Germany, and in Spain. And people realized that it was attacking the roots, and American grapevines actually had complete resistance to this insect. So lo and behold, they grafted American rootstocks to the European top part, the scion, and they were able to form these graft combinations that were resistant to the insect but could save the wine industry. So nowadays, the vast majority of grapes globally are being grafted for this reason. So when we go back to my specific research, I have a lot of questions that we're, of course, very interested in. We want to try to understand, why do grafts actually succeed? And why do they fail? We want to understand, how do plants graft? And we're interested a little bit in these processes you see here. You see these natural tissue fusion. Is this related to grafting? Is this the same process, or is this something quite different? We want to understand how we can try to improve graft junction formation, so try to get new combinations and the existing combinations to work better. And we're also very interested in parasitic plants, this so-called darker side of grafting, where again we have two plants joining together, but in this case, like the example of mistletoe here on the right, you have an interaction that is actually can be quite negative for the plant. So the research that I've been doing in my lab over the last 8 years here at SLU has really focused on using the so-called model plant Arabidopsis, or in Swedish Backtrav. And this plant, we graft it when it's extremely small. So these plants are only 7 days old. I graft them and my team using microscopes. And you can see the graft junction is actually just at the top here. We cut it right through this kind of embryonic stem and we rejoin them. This is a very efficient process. It goes very quickly. We can have nearly 100% success. We can make almost 80 grafts per hour. And using this, we've made a lot of fundamental discoveries about plants and how they graft. We've discovered cell types, we've discovered genes, we've discovered proteins and pathways that are important. We can also use microscopy because these tissues are so small, we can put them under a microscope and we can create beautiful images here on the right where you see the different colors coming together as cells divide and genes express. So we've taken fundamental discoveries with this Arabidopsis, and now I'm going to try to present a couple examples that are slightly more applied that we think could be of relevance in the future. And one of these is actually looking at the monocots, and this is a very important group of plants. This involves the cereals, so barley, wheat, oat, and rice, some of the major crops in the world, are monocots. And they are considered to be ungraftable. So you can see an image here on the right that I tried to graft many years ago, and it's a disaster. They cannot be grafted. And we think there's two issues. One, they lack stem cells in their stem, and they also have their vascular bundles scattered. This is our monocot here. So they're kind of all over the place and a little bit randomly scattered. So this was a major issue. But what we realized, us and our collaborators in Cambridge, is that we could actually use extremely young tissues. So if we took seeds that were only 1 day old, this is the technique here. You take a seed, this is the endosperm, this is the nutritious part, this is the embryo, and we can actually pop out the shoot of the embryo, and we can replace it with another plant as long as that seed is only 1 day after germination. We let these plants recover, and they actually successfully graft with fairly high efficiency. And you can see an example here where we have our rice graft, which is formed. The junction is here where I'm pointing. And here we actually have some different colors that have come together. So we have a blue plant with our white plant. And again, you can see the graft junction. And we can also do this for other monocots, not just rice, but it works for wheat. There's an example here with date palms, and we can also do this for with bananas. So normally when we graft, the species have to be quite closely related. What was very surprising with monocots is suddenly we could start grafting species of monocots that were very distantly related. So by up to 50 million years of evolution, we grafted a cereal known as pearl millet to wheat, wheat to another cereal known as sorghum, and rice again to sorghum. And these grafts were quite successful, so it was extremely surprising. Is there any sort of relevance to this? Can we take this forward to some sort of application? Potentially. And one thing that we looked at, and our collaborators in Cambridge, is that they were looking at a fungus disease called take-all disease. And this take-all disease badly affects wheat. So you can see these wheat plants are dead, they've turned black, and they are very unhappy with this fungal disease. Oat is highly resistant to this disease that attacks the roots. So when we graft a oat root to a wheat shoot, as you can see over here, the plants become completely resistant to this fungal disease. Another example that I would like to talk about is a little bit about Swedish forestry. And of course, forestry is incredibly important for Sweden. It contributes something like 10% to the economy of Sweden. There's a couple species that dominate. One is spruce and the other one is pine. And grafting is being done commercially in Sweden. There are several thousand trees being grafted each year. And they are primarily grafted for clonal propagation to make more copies of something that's very, very good, being done in areas known as seed orchards. These are parts of Sweden where they have land where we have very, very good trees growing to produce millions and millions of seeds that are needed for the forestry industry every year. And of course, we're very interested in using grafting to see if we could not only improve growth of the trees and to modify their growth, but to also see if we can improve disease resistance. So if you recall earlier, I talked a little bit about Arabidopsis, grafting with very young tissue. I talked about monocots and grafting with very young tissue. And we tried the exact same thing with conifers, with our gymnosperms. So we took, instead of using traditional grafting techniques, like you could see here, this is done on several-year-old trees, and we're looking at about maybe 100 grafts a day. With our grafting techniques, we again used 10-day-old seedlings. We cut them through their sort of stem here. We switched them around. We put on a little collar to help them reconnect, and we can increase efficiency, or the number we can do, by about 4-fold. So now we can graft 400 a day, and we have very high success rates. So that was very nice. We started to look at grafting different species together. And when we graft our pine to itself, it works very well using our micrografting, or very young grafting. We did another pine species. This is lodgepole pine. Worked very well. We could now graft lodgepole pine together with Scots pine. So it's the same genus, but a different species. This worked very well using our micrografting. But what we also discovered is that we could suddenly, using this very, very small grafting, we could actually graft pine and spruce together. So here we have a pine shoot and a spruce root. So these plants are 3 years after grafting. So they were grafted as very young seedlings. 3 years later, we can now graft spruce and pine together, which is something that would normally be impossible to do. It hasn't been done before using more mature trees. And finally, my last example of grafting here that I'd like to present is talking a little bit about working with another species, and this is with tomato plants. And tomato is very interesting. You can form nice grafts with potato. So here you can see we have a tomato on top, potato on the bottom. You can buy these commercially. They're called tomtato, and you can understand the benefit. And they graft very, very well together. Another closely related species to tomato is pepper. And tomato pepper grafts are a disaster. So when we graft these in the lab, you can see them here. Pepper grafted to tomato, the plants typically die and fall apart. So even though these species are very closely related to each other, the grafts fail in a very spectacular way. So now we're identifying the genes and the pathways involved that we can actually rescue this graft junction formation and try to apply it to other species. In the final part of my talk, I'd just like to talk a little bit about parasitic plants. And these are very important from an ecological point of view, but also from an agricultural point of view. They cause billions of dollars of damage every year. The largest flower in the world, Rafflesia, is a parasitic plant, but we also have examples like mistletoe here, which is infecting a grafted apple tree. So you could imagine there's 3 different plants here. We have other plants like dodder, this spaghetti-like plant that's covering and infecting our bean. And we're very interested in parasitic plants because we think there's a lot of parallels with plant grafting. You can see here, this is our grafted plant, our shoot and our root, and you can see this beautiful green color of the vasculature connecting between the 2 halves. The plant parasites do something conceptually very, very similar. We have our parasite here on our left, our host on our right, and in blue you can see the vascular connections that form between 2 different plants. So we want to know how parasitic plants can do this, and they can also infect very distantly related species, something that's generally impossible to do with grafting. So can we take the knowledge from parasites into grafted plants? And it's just one quick example of some of the research that we've been doing. We've been working a little bit with a species called striga, known as witchweed, which is incredibly devastating in sub-Saharan Africa and Southeast Asia. And we don't work with striga specifically in our lab, but we work on a close relative. And rather than doing field studies in Africa, we do a lot of our work in the lab, and we set up these sort of in vitro assays where we have our parasite, and we can infect it with hosts, and we can see how the system actually behaves under very controlled conditions. And what we realize is that actually nutrients has a huge effect on our amount of infections that occur. So when we give high nutrient levels, both actually to the striga system and also our parasitic plant, we get a very low number of infections. When we starve the parasite, we get a much, much higher level of infections. So you can see here. So what's happening is it looks like the parasitic plant is monitoring nutrients in its environment, and it's adjusting how much it affects according to that. So with that, I'd just like to come to the end of my talk. I think the future of grafting, of course, is very bright. This is becoming extremely popular and it's growing. Our knowledge of grafting itself is also growing rapidly. We have a lot of genes and pathways of knowledge that my group and our collaborators and others in the field have contributed. We have new combinations of plants that are emerging, and we think ultimately we can create plants that are more climate tolerant, more disease tolerant, and can grow in new environments. We can also improve yields and also modify flavors of plants. And ultimately, the parallels with grafting and parasitic plants I think are extremely interesting, and we have a possibility to exchange knowledge between them. So with that, I'd like to thank you for listening, and I'm happy to take questions probably after my discussion with Malin.
Thank you for a very interesting talk. You're welcome with questions, and those of you that participate online, you can send your questions to nyaprofessorer@slu.se.se. I have a question. You mentioned micrografting in cereals and conifers, and how realistic is it that this could work in the future?
So I think the technique, works fairly well for us. So we have good success rates. We can do several hundred grafts per day. I think one of the real limitations will, of course, be the number of plants that's required. So in conifer trees, we need 400 million plants a year. With cereals, it could be in the billions. And when you have a very high cost of grafting, it just doesn't make sense to graft wheat plants or rice plants that are very short-lived plants. If you have a tree that's going to live for 100 years or 120 years, and you can make it more climate resistant or disease resistant, or you can improve its growth by 30%, then it might make a lot of sense to pay that extra amount to have a grafted plant that's going to be much more tolerant or yielding.
Okay, we'll see. Any questions? Not yet. Not in my phone either. Well, what do you think about? Well, you mentioned that certain plants are easy to graft, and others just don't work. And why is that?
So there's a couple different factors here I think at play. One, I think, is grafting is almost a bit of an art form. And you have to pick the right time of the year, the right species, the right technique, and you have to be very good at grafting. So I'm quite good at grafting in the lab. You can see there was this image I showed of Arabidopsis, but in my own garden I'm very bad at grafting.
Okay.
It does not go well for me.
Okay.
So of course, the people who are professional grafters, they know exactly what they do. They have very, very good success rates. But of course, that's only part of the story. I mean, there's a lot more going on. So there's some plants that are very easy to graft, like apple trees. So if you're going to start grafting on your own, you might want to start with apples. There's other plants like monocots that are a disaster to graft. And this probably has to do a bit with their ability to regenerate. There's some plants that are just naturally better at healing. And there's also probably something to do with how plants recognize each other, because there's certainly some examples where we see graft combinations work initially, and then with time, things really start to go badly.
Okay.
So there's very, probably species-specific reasons why things don't work.
Yeah, and just to come back to the apple tree, what is it really that makes it more easy to graft the apple tree? Maybe you mentioned it, but just
it's a good question. I mean, I speak to commercial grafters, and when they compare cherry trees to apples, they consider apples the easier one, whereas grafting cherry trees is a little bit harder. But if you looked at them in your garden, it wouldn't be obvious what would be the difference.
Okay.
But it could be the tissues, the anatomy, and this ability to heal.
Yeah, and what are on the more personal level, what are your experiences from grafting in your garden? Have you succeeded in any cases?
So we can talk about the grafting in the lab, and there I have good success. There's many species in my own garden. I'm grafting apple, cherry, and peach. And it's for the most part the success rate hasn't been particularly high, but I think this year I'm optimistic.
You're working on it. Yeah,
I've got some professional advice a couple of weeks ago from a professional grafter who showed me some nice techniques.
Okay.
So it's
good luck.
Thank you.
We have a question over there.
Thank you for a really interesting talk. Wow, you are. I get worried about my apple tree now because I'm hoping that it will work out fine. My question is, do you know of any grafting within forestry on large scale that has happened?
Yeah, so it's an interesting question, because for me, forestry would be an obvious application for grafting, because the trees are extremely long-lived and you have this sort of potential to, I think, really improve a tree. Within Swedish seed orchards, so these are distributed all over Sweden, there's a lot of grafting that's being performed. So perhaps we're talking about 10 or 20,000 trees a year in Sweden that are being grafted specifically for seed orchards. And these are the best trees in Sweden. They are genetically extremely good and they want to clonally propagate them. Outside of these sort of seed orchard environments, then grafting, as far as I'm aware, is not particularly common. But, within the seed orchards, I mean, you can do things like you can graft to accelerate flowering. We're very interested to try to make the trees smaller in seed orchards to actually cause dwarfing. So what I presented with the apples, if we could repeat this with our conifers to make them flower faster, more flowers, get the trees smaller, could be a big advantage for the seed orchards.
We have another question.
Yeah, thank you again for a very interesting talk. Can you explain a bit more about the differences in the reaction of the immune systems if you compare plants and animals? I don't understand that.
Yeah, I mean, it's so I'm a plant biologist, so I can certainly talk to you about plant immune systems. And plants have something called an innate immune system. So they tend to have receptors that are looking for something and then they activate. And when we have a graft that succeeds, we have very little immune response. So it seems that everything is fine. So even if the plants are the same species or maybe a little bit distantly related, it seems the immune systems do not react. When we have a graft that fails, so maybe an example would be the tomato pepper that I showed to you earlier, there seems to be some kind of immune response. But we're not sure if this is a cause of failure or it's just turning on later because things are so unhappy and not connecting well. At least with, when we talk about animal immune systems, they also have this innate immune system, but they also have this kind of reactive immune system. And the immune system, of course, is extremely sensitive. So if you give a blood transfusion and use the wrong type of blood, if you don't match the blood groups, it can be a disaster. So they are, the animal immune system is extremely careful about foreign objects or even within the same species. But plants, we don't seem to have that issue.
It's so interesting that plants can't, separate friends from foes. They react in the same way when grafting. Yeah, that's what you
that's right.
talked about. And I think that's quite strange in a way. But well, anyway, next question.
Okay. Thank you, Charles, for your presentation. You started talking about your first studies was on Arabidopsis, or at least in your presentation, and I think partly that was because Arabidopsis is very well known in terms of the genes. How important is the study of the different genes and the activation of genes for your work? Can you tell us a little bit more about that?
Yeah, so our research on Arabidopsis, I think, has been absolutely critical for the progress that we've made in other species. We spent a long time figuring out what Arabidopsis is doing, the types of genes and pathways that are being activated, and we can describe genes that improve graft formation and then block graft formation. And we can also describe conditions that can also affect this. So like temperature, we discovered that if you increase the temperature to a certain degree, you get much better grafting success. And because Arabidopsis is so quick and easy to graft and we have such good tools, we can make very, very rapid discoveries. And then the idea is that we've taken these discoveries and then now we apply them to the monocots. So we've also tested some of these things in monocots. We've also tested them in tomato. And then this allows us, and for the most part, it's actually been very consistent. So it doesn't seem that different plants have developed different ways to graft. They're probably doing something very similar. Even our spruce trees, we've compared them to Arabidopsis and they're activating the same types of genes. So this has been a huge advantage to us. I think if we started working with apple trees and really focused only on apples, we would have had a much more limited understanding because working with apples is so challenging.
We have another question.
Okay, thank you. A question about graft. Sometimes there are great problems with graft incompatibility within species. Can you say something about that and what maybe could be done about it? For example, Douglas fir is one of these species that is extremely hard in this case.
Yeah. I mean, it's a really interesting topic. So I've talked about how closely related species generally graft, well, and this is true for a lot of different species, but then we also have examples like Douglas fir. We also have examples like London plane trees, which are sort of a type of sycamore. And again, where you maybe have 20 different individuals, one is extremely good at grafting and the other 19 are extremely bad at grafting. And it's a real mystery how, it's sort of it goes back to why would you have one plant that doesn't seem to mind the others, but then the others really have an issue. And at the moment, we don't know. And I think part of the challenge is because it's Douglas fir, because these are London plane trees, it's very hard to actually figure out what genes are involved in this process. If I had to guess, I think it probably has something to do with their hormone response. And they're probably, there's one tree that just seems to be, a universal donor that doesn't seem to mind who it's actually being connected to. But the other ones are having some major issues. They're having an immune response or there's something to do with their hormones.
Thank you.
So when you talked about the striga example, you referred to high nutrient levels making the striga less sort of prone to attack the maize or sorghum.
Yes.
But I've heard this being described from the maize breeder point of view, and they talk about it as high nutrient levels making the host plant more resistant to the attack of striga. What would be your comment on that?
Yeah, so I think that's very true. So we think there's probably at least 3 things going on here. So it's fairly well known that in a nutrient-poor field, there can be very, very high load of parasitic plant infection. So one thing the farmers can do is basically treat with synthetic fertilizers or manure or whatever to reduce the amount of striga in their fields. This is a huge benefit. And what we think is happening is the first thing is probably what you described. The nutrients are making the host plant stronger, but they may also be making the parasite stronger, but there's some evidence it makes the host stronger. The second thing it does is it actually causes the host plant to release less strigolactones, so less hormones in the soil that cause the striga to germinate. So this is a little bit, I didn't cover this. The third thing that's going on is it seems that the striga is actually forming less infections. So this is the data that I've shown. So I don't think there's one that's dominating, but we think that probably all 3 things are happening and contributing to this phenomenon.
Then one last question. Looking ahead, what's the big dream for using grafting as a tool in plant science and farming?
So I think within plant science, I would love to really understand why some grafts fail. And I think some of the examples we talked about today, I showed you a lot of examples. We can graft very distantly related things, and that's very interesting. But I'm really interested why very, very closely related species cannot graft, or even within the same species. So I would love to know what are the receptors, what are the immune system, what's happening. Within agriculture and forestry, I think I really like this idea of combining the best of 2 plants and then taking this forward to make sort of a superior plant. And I think nowadays people are getting very interested in perennial crops, perennial cereals. So maybe there's a applicability to this. I would love to lower the costs of grafting and also make it, more accessible and more widely known so that, farming and agriculture can take a benefit of this.
You have a long to-do list.
Yeah.
Yeah. Good luck with that, and thank you very much.
Thank you.